Okay, class. I'm going to continue on the lecture 15. This is slide number 12. Another another time to showing this the gradient of the nuclear dorsal protein. I think that's showing the fate map over here. It is showing nice ventral, distribution, and that is contributing to this, fate map over here. Question is, dorsals are not in this dorsal area. Okay, okay. So they are not in dorsal. And then dorsal has a particular structure like this, okay? So how they are going to make this kind of structures? So we have the particular signaling that are mostly like a toll signaling, right? Okay, in ventral side, okay? But how about on dorsal side? What is really going on? So this is kind of like after story. It's really hard to kind of like draw a clear line when is really occurring and in what order. Okay, so because people has to look at the one thing at a time and really didn't have a much chance to take a look at you know uh, two events at the same time. So this is from your book. I think that try to be more focusing on the dorsal side because the in here toll signal is dominating. Okay, but the in here because of that there's no signal is another signal. So totally different set of signaling will be uh, be turned on. So that is, I think that's, I wish you can read it here, decapentaplegic uh, DPP. So decapenta means the 15, and plegic means the defect or like a phenotype kind of thing. Okay, so it has a 15 different phenotype. So DPP will be expressed on the dorsal region. Okay, so here's the DPP, area, and then DPP will be, distributed around this dorsal hemisphere. So DPP and then toroid protein will be on here. So toroid and then DPP will be on this dorsal hemisphere at the same time. And I think that there are some other protein that is SOG, okay? And SOG is laterally expressed. And then what the SOG does is we need to look at this, diagram really carefully. So SOG is like a blocking the function of DPP. So it's kind of like a dam or blocks that, okay, suppress the DPP activity on the lateral or ventral side. So that's how they can have the final nice balance. In dorsal, they have this DPP signaling is active, and then ventral toll signaling is active. Okay. So, we'll go back to the ventral cells. And then in the ventral, uh, we know that dorsal is the ventralizing cell. And then the one of the target, the major target of the dorsal is twist. Okay, this is also a gene name. So this is showing the uh, twist expressing cell, and twist is being almost like a we call as a mesoderm marker. Okay, So they produce cells that producing twist will become mesoderm. So they will be forming, undergoing invagination, and then they will be, forming a tube, and then it will be internalized, and then collapse those, and then probably those cells, migrate to the many different places. Okay. So this is the mesoderm marker, and then I would say it's a dorsal target. Okay, right. So gene expression pattern, responds to this dorsal gradient. So in the ventralized cell, this is like a ventralized mutant, isn't it? I'm showing up here. Okay. And when dorsal is active, then what will happen is that because of that, twist will be turned on all around. So that's why they become all the ventral structure showing up. And they are nicely balanced, and dorsal will be only on the ventral. So only on the ventral cells produces this activate twist. So that's why they will have this nice ventral axis formed, and that is probably the kind of like a certain level of the twist and dorsal will be determined, balanced. Okay, right. Okay, so if you look at the dorsal gene, then what will happen is that dorsal will not only activate twist, but also they will activate some lateral genes such as rhomboid and the snail. And those genes are actually the snail and the rhomboid also, they have some in the interaction. I mean, I think this really important thing in here is this, okay? Dorsal is activating twist, okay? Okay, and we'll go back to dorsal, and then dorsal, what will happen is that this DPP, decapentaplegic, is the TGF beta family protein, which is the BMP4, and this is the secreted protein. Okay, right. So this secreted protein, will be secreted out, and then probably they will be, going into perivitelline space. Okay, right. And then I think that's, there will be a spread over. So what this decapentaplegic does is that, okay, so, dorsal cells are here, okay? They will produce this, all around, okay? They will produce this toroid, decapentaplegic together. Zirconalt is kind of like a kind of a dorsal version of the dorsal side gene. It's a dorsalizing gene, okay, right? So, so it's upstream of the DPP. So they will be produced, and then what I'm trying to say is that, so for example, let's say you secrete something to the outside, and then you receive that signal back, and then you, differentiate into something else. It makes sense, okay? So it's like a self, stimulation. So let's say you have a cell here. Let's say I would just say just layer of cell here, and then they'll produce protein. Let's say something like this, okay? And then this is the some kind of growth factor. I mean, if you want, you can call it a DPP, okay? Right. And then these cells, these proteins, is coming back to you, coming back to this cells that produce this cell, and then undergo some kind of changes. It could be a similar thing is, we call it an autocrine signal is like you know, self stimulating. So bottom line is dorsal cells in the dorsal area produce DPP, and then DPP is produced and then coming back to them again and then turning into a dorsal structure. I wish we could have the much better diagram or something. I tried to find the video or something, but I just could not really find something good. Okay, so what that does is the problem is, okay, let me go back. Okay, so they are produced in peributyllin space. So this is also same problem. So this is the space. So that is all the way connected to the ventral side. And if DPP is produced from the dorsal side, then what will happen is the DPP might as well diffuse down to the ventral side. And then they may change that ventral side into a dorsal, isn't it? Dorsal structure. So that is also a possibility, okay? So so that's why, you know, this SOG, short of gastrulation, I think that is the SOG, okay? Okay, and the function is really important. And then SOG is, you know, expressed in laterally over here. So it's very high. I think that's high, you know, somewhere here, okay? So so that's why, you know, although DPP is produced in the on the dorsal side, and then they may diffuse down to the ventrally, but SOG is blocking. Okay, SOG is blocking the function of DPP. So this is different version of the same book, and SOG is the blocking it. So what will happen is that's how you know, DPP can form the gradient. I mean, SOG is also secreted protein, so they can also go all the way up. Okay, right. So usually they will also form a really gradient. So on the top, SOG is very low, and then DPP is very high. Okay, so that's why they can form this kind of a nice little gradient, very, very high dorsal activity. And then as they go to the laterally, they will have a less and less dorsal activity. So that is the problem. One thing that I forget to mention is that, okay, so from here, isn't it, SOG is also secreted protein, and then SOG may, you know, diffuse all the way up and then wipe out the DPP function, isn't it? Because, you know, I mean, might as well they can straight up. So that's why let me use the different color here. Toroid, okay, toroid is also right over here, either same as this DPP, so that toroid is also killing the, you know, toroid is blocking the SOG activity. I think this is like an endless game, you know. It could be you know, some different way of explaining things, and then, you know, I'm not the you know, fly you know, biologist, so I'll just bring it up to you at this moment, okay, up to here. I mean, then you can ask, you know, how can we control toll? I think the toll is somehow tethered to the eggshell or something. But other than that, you really, I think that's probably that's out of my ability to figure it out more. Okay, right. So anyway, you know, it's kind of complicated, but this is how they can maintain the gradient of DPP. So you know, this is from a different book. Okay, and I copied from you know, the previous version of this you know, MBC, which is the molecular biology of the cell. You know, it is showing much better you know, representation. So dorsal is expressed like this, right? So because of that, the is created. The SOG will be expressing laterally, and then here's a twist. And then DPP will be expressed. So SOG is also, secreted out, and then DPP is secreted out. But here, the reason why the DPP will not come down is that because of the SOG. So, one thing I want to mention is that DPP is almost similar to SOG. What do you mean is that the structure. Okay, so they are almost same identical protein. And then what the SOG does is that is 100% of their purpose, their function is to block DPP. So all they do is to block the DPP. SOG does not have any receptor to bind to the on the surface or something. Okay, cell surface. So because of the interaction between DPP and SOG, basically can form this nice little gradient. And then because of that, they will form this different, I would say, patterns based on this balance between, SOG, DPP, and then bottom here, that is the special. Okay. Okay, so next is what we are going to talk about is the, we're gonna switch topic to amphibian Xenopus. And then we are going to talk about this mesoderm induction, okay? So, fate map technology, it is showing here, kind of like a dye, that fluorescence dye, they stained over here, and when they follow through, and then they are going to this mesoderm. Okay, so that's how they found this area. They are forming the mesoderm. I think that's they took the area a little bit deeper, I guess, right? Okay, so those fate map, and then what they found is that mesoderm is formed in the middle. Okay, and then also not only just formation of this three germ layers, but the mesoderm forms pattern from the dorsal to ventral. So in dorsal, they are forming notochord, which is the spinman organizer, and then stomite, and then mesenchyme forms in ventral side. And also, ectodermal cell has pattern, has the, I think the ventral side is epidermis. And then, dorsal side is, a nervous system. And that is the some kind of patterning of this Xenopus really going on. Okay, so the cell movements of the during the gastrulation, so they'll form. Gastrulation is a cell migration in the formation of the organ process. So after they form like this, and then they migrate, okay, and then they'll really live up to their names. So endoderm goes to the center, and then ectoderm goes to the outside, and mesoderm goes to inside of the in between. Okay, right. So how those different germ cell layers are specified, especially the that is the mesoderm induction. So how they determine induction. And then also you saw the pattern, isn't it? Those are to a ventral pattern. Wow, notochord to the mesenchyme. So we're gonna learn these two things, and then these two, how these two are really important in formation of the dorsal and ventral axis. Okay, and then later on gastrulation. Okay. Okay. So I'll continue that in next segment. Thank you.